Our course title is Microbial Technology. Lecture title is Genetically Modified Microorganism. Topic discussed in previous lecture. Detoxification of pollutant by bioremediation approach are varied and have been successful in cleaning up a location and have been contaminated by various pollutants. Since microorganisms are essentially to bioremediation, information about their diversity, abundance and the community structure is in contaminated environments can be obtained by monitoring other environmental factors that may hinder microbial activity and ensuring that they may remain within an optimal range. Characterizing the site in the first step towards a successful bioremediation, since it helps determine the most appropriate and practical bioremediation process in an ex situ remediation. In situ approaches, on the other hand, do not additional costs related to the excavation, whereas nevertheless certain in situ method of bioremediation may be ineffective due to the high cost of equipment installation on site and the inability to properly visualize and manage the subsurface of pollutant site. The added expenses associated with excavation and shipping. Ex situ bioremediation process are typically more costly. However, they can be applied to a variety of contaminants in a regulated way. Topic to be covered in this lecture is introduction, reason for the genetic modification of microorganism, tools and techniques for genetically modification of microorganism, basic strategies or approaches to produce product yield, genetically modification of microorganism, then application of genetically modification of microorganism, bioethical amplification of genetically modified microorganism, risk posted by genetically modified microorganism, risk monitored of genetically modified microorganism, summary, conclusion and references. Introduction. Genetic engineering or genetic manipulation is the technique of altering an organism sequence of nucleic acid in a lab setting through the addition or deletion of the base pair, deactivation or inserting a virulence gene that is not needed or producing a new trait in genetically modified microorganism. Historically, humans have altered microorganisms to create delicacies like wine and bread, but today industrial and medicinal uses of genetically modified microorganisms have been done. Saccharomyces cerevisiae and lactic acid bacteria are the example of that is most common by perform chemical transformation, then help produce metabolites of commercial or medical importance such as insulin, vitamins, amino acids or enzymes. Most reasons to prefer microbes because microorganisms grow rapidly and in most cases are easy to cultivate. Using microorganisms in much environmentally friendly than conventional chemical synthetic methods, they use less energy and use renewable sources. Today, genetically modified microorganisms, which is called GMMs, have found application in human health, agriculture and bioremediation and industries such as food, paper and textiles. Genetic engineering offers sufficient supply of desired products, cheaper productions and safe handling of otherwise dangerous agents through product of GMM have largely been accepted and consumed by the population. Their utilization remain questionable as regard the role and implication in the ecosystem. This has raised ethical concern relating to dignity, respect for persons, consequences and justice of their utilization and acceptability. Now, the reason for genetic modification of microorganism. Generally, traditional biotechnology made use of microorganisms such as yeast in baking and lactobacillus in the production of dairy products. Biotechnologists were faced with so many challenges using the wild microbial flora. These wild microbial floras were usually unstable and not consistent in their ability to produce the desirable product. it becomes very difficult to biotechnologists to obtain a homogeneous product within a long period of time, and therefore was unable to manage the quality of their products. The microbes sometimes were unable to survive in the fermenter. The condition within the fermenter inhibited or altered the normal functioning of the microbes and their survival. Also, the final products were even toxic to the microbes due to the feedback repression. Industries in these domains were usually faced with the low productivity, probably due to the low growth rate of these microbes. These low growth rate of microbes made the cost of production expensive. All this difficulty faced greatly affected the output of these industries financially and economically. There became need to stable microbes with good resistant fermentation condition and enhance productions. With the discovery of genetic engineering, biotechnologists find out these microorganisms could be genetically modified such that they would resist fermentation condition. For example, cloning a gene which encodes for thermoresistance genes to survive the high temperature in fermenters. Genetic engineering could also produce clone of the particular microorganism which is stable with similar properties and could multiply rapidly. With all these advantages, genetically modified microbes had an argue over wild flora and were rapidly adopted in biotechnological purposes. Tools and techniques for the genetic engineering of microorganisms. To manipulate microorganisms for the expression of desired traits. First one is gene transfer method to deliver the selected genes into desired host. Second, cloning vectors. Third one is promoters to control the expression of desired gene. And the fourth one is selectable marker genes to identify recombinant microorganism. First, gene transfer method. Transformation in this process uptake of the plasmid DNA by recipient microorganism is accomplished when they are in physiological states of competence. Electroporation is second one, alternative method to transform DNA into microorganism. In electroporation method originally used to transform eukaryotic cell, a high voltage pulse make recipient cell electrocompetent. Then transient pores are formed in the cell membranes as a result of electroshocks, thereby the allowing DNA uptake. Third one is conjugation. This method involves a donor strain that contains both the gene of interest and the origin of transfer ORI-T on plasmids and the gene encoding transfer function. The contact between donor and recipient DNA transform occurs. Vector are cloning vector. To carry out genetic modification depends upon choice of the transport. Then next one is the promoter. Promoter is segment of the DNA that regulates the expression of the gene under its control. Promoter are various types which we use in genetic modification. First one is constituent promoter, which is used continuously active, and inducible promoter activated only certain condition. Presence of inducer selectable marker genes which often encodes proteins conferring resistance to the antibiotics for identifying the transformants. In this way, genetic engineering frequently makes use of a variety of transformation, conjugation, mutation, electroporation, molecular genetics, recombination technology and the protoplast fusion. 3 gene editing tools have been developed in the recent year using a combination of genetic engineering and biotechnology. These includes transformation associated recombinants, which is also called TAR. Second one is transcription activator like effector nuclease, which is also called TALEN technology. Next one is zinc finger nuclease, JFNS technology, and last one is the clustered regularly spaced shorter palindromic repeats, which is also called CRISPR associated or CRISPR-Cas technology. This is the figure by which basic recombinant technology is done for the formation of genetic manipulated microorganism. These technology are actually the 3 generation technology and allow gene editing for the breaks in both strand in desired gene sequence. The development of the genetic tools, techniques and genomic sequencing has made it possible to introduce new strains of microbes that are used not only for diagnosis but also for the prevention and treatment of diseases. This is the diagram we show. First part is showing genetic modification technique, which we already deal, and the second part is showing part B, genetic engineering or modification outcomes or bioengineering or traditional breeding occurring in nature by which we form genetic manipulated microorganisms. Next is the basic strategies. Strategies for the genetic engineering of microorganism are: first one is description or complete removal of undesired gene or their pathway. Second one is overexpression of target gene. High expression is an drawback. It leads to segregation of the desirable genes and losses of desired traits. Then improving protein production properties. For these strategies, approach to enhancing the product yield are overcoming rate, that limiting steps. Then the second one is eliminating feedback regulation, and the third one is manipulating regulatory genes. Genetically modified microorganisms, which we deal now. First one is genetically modified bacteria. Many genetically modified bacteria for use in the environmental protection, disease treatment, food industries, chemical synthesis, etc. have been developed after decades of work. First one is showing bioremediation, the production of the bacterial recombinant system that may break down different types of pollutant. First pollutant is hydrocarbon removal. In 1971, the Chakraborty's group generated the first genetically modified Pseudomonas bacteria and had the ability to break down the 4 different hydrocarbons of crude oil. The second one is metal contaminant removal. In this, mercury may be effectively removed from electrolyting effluent by genetically modified E. coli that simultaneously harbors the MT and the MRT and the MERP genes. Second one is the removal of cadmium. It was developed to use genetically modified E. coli JM109 strain or Pseudomonas putida, Colobacter species and Mesorhizobium hawkei for the cadmium bioaccumulation from a heavy metal contaminant site by expressing protein fusion for RSA A 6 Hayes recombinant protein system, phytogelatin PC and the metal binding peptide EC20 respectively. The third removal is arsenic. High capacity for removing arsenic from hazardous location is also promised by metallar regulatory protein ARSM and ARSR gene expression in E. coli. Next one is the removal of dye. A genetic engineered bacterium was created from a strain of Burkholderia cepacia to digest these pollutants. This was initially studied in the US for the cleanup of Agent Orange. Then fourth one is the pesticide removal. Recombinant strain of Sphagomonas species strain CDS1 created for the organophosphate and carbamate degradation. Then Harni Samueli School of Engineering and Applied Science produced a genetically modified microorganism E. coli for biofuel synthesizer. Next one is the application in the field of medical. First, for immune protection or vaccine development, Salmonella, Neisseria meningitis, Listeria and Vibrio coli are a few examples of the genetically modified bacteria that can trigger in vivo immune protection responses by inserting certain potent antigens into low or no virulent microorganisms. Then production of the recombinant vaccines. Saccharomyces cerevisiae, a common Becker's yeast, produce hepatitis B virus against vaccine based on the hepatitis B surface antigens, which name is Antigerix B. Recombinant vaccines to eradicate rabies, anti-rabies gamma globulin treatment. Second one is the use in cancer therapy. Certain bacteria like Salmonella, E. coli and the Clostridium novae anti-strain have a unique capacity to colonize solid tumors, making them great candidate for drug delivery system in cancer therapy. For instance, S. typhimurium means Salmonella species that express alpha pseudomonas exotoxin, a transformed growth vector, showed notable growth inhibitory effect on MC38, 4Ti and CT26 solid tumors, whereas PD1 siRNA and CpG ODN activated innate immunity and suppressed PD1 expression, which killed cancerous cell. Next, use in textile industries. Alpha amylase of the Bacillus species have been used for many years to remove starch size from fabrics known as daisy. Originally, amylases from the plant or animal source were used. Later, they were replaced by the amylases of bacterial origin. Next one is use in the field of agriculture. Nitrogen fixing genes like NIF-L and NIF-A can be inserted in Rhizobium miloti strain of the bacteria, thereby increase the amount of nitrogen fixation by these bacteria. Indigo, a commercial blue pigment that is used to dye both cotton and wool, was originally isolated from the plant, whereas recombinant E. coli bacteria with plasmid NaH7 is used for producing indigo by biotechnological method. Next, use in as a insecticidal product. Toxin gene from the Bacillus thuringiensis is inserted into Syncytius and Syncoccus species photosynthetic cyanobacteria that are food source of the mosquito larvae. Coccobacter crescent and equitim bacteria, the insecticidal protein is highly toxic when ingested by mosquito larvae. Next one is genetically modified fungi. It is also used in the bioremediation. Dioxin can be broken down by the recombinant Saccharomyces cerevisiae cells that express the mammalian CYP1A, a complex of the cytochrome P450 family gene with yeast NADPH P450 reductase, which has greater metabolism of tri-CDDS and DCDDS and MCDDS, and the DD that those of the naturally occurring microorganism. Then Fusarium solani, RNBP link KL mixed genotype recombinants were breed with enhanced dehalogenase activity demonstrating better DDT biodegradation capability. Next, bioremediation of chromate, a methylotropic thermotolerant recombinant strain of yeast, that is Hansenula polymorpha, was employed. This strain produced the enzyme F1 avocytochrome B2, which is also called FCB2. Furthermore, is overproduced and utilized in the development of cell-based biosensors. In the field of medical science, fungi are an excellent producer of naturally occurring secondary metabolites with therapeutic qualities such as alkaloid, polyketides, isoprenoids and non-ribosomal peptides. These compounds can be modified to replace them to produce a desired secondary metabolite. Genoderma lucidium and the Saccharomyces cerevisiae share the CRIP gene. This synthesis of novel ganoderic acid with antibacterial action 328 dihydroxy lesnosota 824 diene 26 oic acid derivative increased by 8%. Next, use in the field of food production. Cheese making to produce chymosin rhizomucor mebai, Endothea parasitica, Rhizopus pusillus, Aspergillus niger, Clubomyces, Lactis, E. coli, recombinant chymosin developed in 1981, approved in 1988. 90% of cheese prepared by these genetically modified bacteria as well as fungi. Commonly used microbes which is genetically modified bacteria and fungi are Streptomyces, Bacillus species, Corynebacterium species, E. coli, lactic acid bacteria species, then Pseudomonas species in case of bacteria, whereas in case of fungi, Saccharomyces cerevisiae and Aspergillus species are used mostly. Genetically engineered virus, influenza, AIDS and smallpox are just a few of viruses that have greatly impacted human history and are frequently viewed as foes. Viruses are easily manipulable due to their genomic architecture and efficient machineries. When the first smallpox vaccine was created in 18th century, there are early records of its use. Louis Pasteur developed the first rabies vaccine in the late 18s. Later, polio, measles, influenza and rubella vaccinations were developed. During gene therapy, introduce functional gene inside the human. Next is genetically modified probiotics. Both diseases affecting human and animals can be prevented and treated with probiotics. Probiotics beneficially qualities can be strengthened and their potential for pathogenicity reduced through genetic engineering, making them safe for use in human applications including immunization and the administration of target proteins and therapeutics to restrict the growth and provide protection against L. lactis Vibrio cholerae bacteria have been modified to express CAI1 genes. Next one, in short, if we see the all genetic manipulated microorganism is applied in this field. So, we find out the application of genetically manipulated microorganisms are There exist several domains in which genetically modified microorganisms are used and have generally been classified into 5 categories as described by from the Laboratory of Microbiology and Food Hygiene in Rennes, France. First one is in the field of chemical industries producing bioactive molecules, in field of agroalimentary where produce fermented foods, in the field of environment various use in agriculture for the pollution control etc. In the field of medicine produce microbes and substance for the therapeutic purpose, for example live vaccines and pharmaceuticals, and last one is the in field of research gaining fundamental knowledge. Bioethical implication of genetic manipulated microorganisms. The use of genetic engineered and its products is related to so many ethical issues. To explain their bioethics implication, it is necessary to outline the basic principle of ethics. Ethics basically rely on 4 fundamental principles: respect for the person, beneficiary, non-maleficence and the justice. First one is the respect for the person states that we should love the life living to us, implying that each person should enjoy autonomy, self-ruled, by capable to decide the make choice, have respect for the individual, the community and local culture. Also, the dignity of people should be taken into consideration. Second one is the beneficence takes into consideration by physical, mental and social well-being of the individuals and benefits of any activity undertaken by means. It supports the development of the science and medicines and its provision to those who suffer, because we should continue to make life better. Beneficence is based on the belief that all people have a intrinsic motivation to love doing good inside of harm, expressed as compassion. Third one is the non-maleficence, which means do no harm dictate that we should be reasonably cautious about the premature use of a technology and any activity before potential risks are understood. And last one is the justice implication, that individuals should have equal opportunity, there should have a even distribution to benefit and equal exposure to risk. In case of vulnerable groups, special concern and protection should assign. These 4 principles set the basis for the bioethics. A religion defines life as a creation of God Almighty. Only Him is the giver and taker lives. This implies that man in his creation and therefore should not manipulate the creation. Arguments based upon life's sacredness suggested that altering life from violates the will of a creator. Some religious critics perceive genetic engineering as a playing God and object to it on the grounds that life is sacred and ought not to be altered by human intention. The failure of label genetically engineering foods means that persons who follow religious dietary restricted will be unable to ensure compliance and their beliefs. Second one is benefits. The principle of beneficence clear state that any activity undertaken by man for his use or the environmental must be beneficial to him, his community as well as environment. Genetic engineering has been very instrumental in improving human well-being and supplied us with products and alternative illness. Next is drawback of and limitation. Through genetic engineering has been beneficial to human, it however has some negative impact on man and the ecosystem. The use of genetic manipulated pathway may affect human health, causing disease, disturb the ecological balance, maintaining certain species, cause drug and herbicides resistance and lower the genetic and ecological diversity. Certain diseases have as well been created by researchers trying to manipulate microorganisms. This was the case of Dutch disease into a new era while testing genetically modified bacteria in fungi. This violates the ethical rules of non-maleficence by doing harm. However, the acceptability of genetically modified products is judged based on the cost benefits analysis, whereas the benefits outweigh the harms. The possible negative impact of the genetic engineering is explained in detail below on risk of genetic manipulated microorganism. Next one is the justice and equity. Genetic engineering is a technique mastermind by the western world. This part of the world dominates the political, socio-economical and the developmental decision undertaken by the world governing bodies. The implication of justice and equity is implementing this technique and the consumption of its product demands for a rational distribution of the risk and benefit. Apart from direct benefits or harm that may result from genetic engineering, which we have already considered, there is also the problem of how genetic engineering may affect the distribution of social goods as well as political rights. Such issues are often referred to as problem of distributive justice. Risk posed by the genetic manipulated microorganism. Genetic manipulation of microorganism produce variant with altered physiological state. In order to survive and disseminate in their environment, they need to adapt to the prevailing condition. The danger posed by the genetically modified organism is therefore related both to their dispersal into the environment and to their potential for adaptation to a new environment. The environment, health, socio-economic sectors are possible area of potential risk of genetic manipulated microorganism. Risk monitoring of genetic manipulated microorganism. Risk monitoring is thus a post-marketing risk assessment measure. The aim of genetic manipulated monitoring is to detect potential adverse effect of genetic manipulated microorganism and their use on human health and environment, and if necessary to facilitate early and appropriate mitigation action. The first step in genetic manipulated microorganism monitoring is to identify potential adverse effect of the genetic manipulated microorganism in the environment. A main tool for the identification of adverse effect in the formulation of cause effect hypothesis derived from the ERA biosafety research result as well as from the existing knowledge of the ecology and the ecosystem theory. The monitoring should be able to address the most relevant effect of the respective genetic manipulated microorganism and its use. The next step is the prioritized the identified effects and to select the prevalent indicator, parameters and monitoring objects that are appropriate to address the effect or relevant protection targets. In monitoring, step by step as well as case by case approaches should be applied in order to be able to identify all possible effects of the genetically modified products. Now, summary. Perhaps the most significant gift science can give us in the coming century is genetic engineering. Comparatively speaking, it is far more accurate than crossbreeding, which has been utilized for generations to change the genetic composition of the microbes. Steps of the genetic manipulation includes DNA isolation, gene cloning, gene designing, transformation, then screening. Whereas various genetically engineering techniques such as molecular cloning and recombinant DNA technology, molecular manipulation and cellular manipulation are available in genetic modification in biotechnology. New innovation in genetic technology such as CRISPR system and siRNA are helped to expand our understanding and ability to modify the suppress and detect organism at the molecular level. Genetically modified microorganisms have permeated society through a variety of channels including agriculture, health, research and environmental management. Even though genetically modified microorganisms have genetically helped human society, There are drawbacks of genetically modified microorganism manufacturing, which is why it is still a continuous issue in many areas of the world. Now, conclusion. Even since genetic engineering was the discovery of 1970, the world has fully been under the era of gene revolution. Genetically modified microorganisms are widely exploited in so many domains, for agriculture, medicine, industries to be the environment. Their products are great importance to man and its environment. However, their undesirable and ethical implication still remains a major worry to many minds, whether they should be accepted or not. The acceptance of genetic engineering or its products is subjected to an individual's desire and priority after making a cost benefit analysis. Precautionary measures such as risk and monitoring and the management have been put in place by policymakers to ensure the safety of genetically modified microorganisms and their products. The acceptance of the genetic engineering or its products is subjected to an individual desire and priority after making a cost benefit. This is reference regarding this. Topic to be covered in the next lecture is Ames test. In this topic, you will understand about what is Ames test, what is mutation. You will also understand about the various types of mutations and how to Ames test help in the direction of mutation. You will comprehend the general procedure of the Ames test by agar-plate technique and its limitation. You will also gain knowledge about the flocculation technique of the Ames test. Thank you.